Hello and welcome to part 1 of my special guest lecture. We're going to be talking about big data, deep neural networks, and translational applications.
Just a brief background and disclosure. I had my bachelor's from University of Toronto in biology and computer science, my PhD at University of Toronto under the supervision of Dr. Michael Failings, looking at neuroscience and bioinformatics, profiling the differences in the injury response between a high-level spinal cord injury versus a low-level spinal cord injury. I'm doing my postdoc right now at the University Health Network under the supervision of Dr. Mohit Kapoor and Dr. Michael Failings, and looking at using deep learning to profile and classify neural stem cells and improving the surgical outcome of degenerative cervical myelopathy. I'm supervised by Dr. Tyrell for my academic teaching, and your anonymous feedback on my lecture would be greatly appreciated. And just a disclosure, I'm the founder of Verismo Apps and Verismo Health. We use machine learning and AI to make applications that range from sales analytics, and we've sold some code over to Terapeak at eBay, and to clinic management using computer vision to enhance patient registration processes and integration into the electronic medical records.
So big data has no singular definition for it, and the definition for big data might change probably a decade from now. But big data refers to data that has immense scale, diversity, and complexity, and requires new architecture and methods in order to manage and extract value from the data. So as technology improves and catches up, what's considered big data today may not be considered big data, say, 10 years from now. Big data is characterized usually by these V's. And there are 10 V's that people talk about, but we're going to focus really on the 4 that's really easy to understand. So volume, variety, velocity, and veracity. So volume refers to data volume, so the volume of data that's being generated. And there's been a 44-time increase from 2009 to 2020 in terms of data. And now we're dealing with around 35 zettabytes of data. If you look at the units here, terabytes is what you generally have in computers now, in all personal computers. And we're quite several orders of magnitude in terms of how much data we're storing. And it's projected to increase exponentially as the years go by. In terms of variety, we're referring to the different formats of data that we're generating today. So we have textual data, numerical data, image, video, and we also have time series data that's collected over a period of time. And there's static data versus streaming or dynamic data. So static data is usually a one-time thing. You generate a document versus streaming data, which is coming in real time, or data that can actually change. A single application can be generating as well as collecting many types of data. So if you're looking at an application like Twitter, you could tweet a text, you could tweet numbers, you could also tweet videos and photos, and all of that would be collected in a single application. In order to extract knowledge from the data that we're collecting, all of these types of data need to be linked together. So a certain user could generate certain data, and you'd be able to associate that data with their demographic profile, and you'd be able to extract some meaning, especially for advertisement purposes, for instance. Velocity refers to the rate at which the data is being generated. So data is being generated very quickly and needs to be processed very quickly in order to get business value out of it, generally. And this really has spawned the age of online data analytics, cloud-based data analytics. So you might think about Amazon AWS, IBM is in the game as well, as is Microsoft. And late decisions in these industries are considered missed opportunities. Like for instance, if you're looking to buy something off Amazon, and you're looking for it really hard at that moment in time, you might find it at Walmart if they don't present you with that particular product in time. So e-promotions is one good example. Healthcare monitoring is another one. So if you have a patient hooked up, there's a bunch of sensors. You'd want any abnormal phenotypes to be caught and reported in real time in order to be able to have the physician immediately act on that. And then the last feed that's less talked about, but it's gaining traction a little bit, is Veracity, which refers to the confidence drop and as data volume goes up. So as you have more data being collected, for instance, we'll talk about COVID-19 data, there's a lot of uncertainty, mainly due to the inconsistency in which the data is being reported in. So you could have one hospital just messing up date of birth, for instance, month and dates messed up, and that's easy to correct. You could also have hospitals that diagnose certain comorbidities in different ways. So if there's no standardization in which the reporting is happening, you could have a lot of uncertainty in terms of the conclusions that you get from your data sets. And this also refers to latency. If the way in which you're gathering the data has a lot of lag, as in it takes a long time for you to report it, then that could also damage the conclusions that you're getting. And a lot of times when you're missing data points, for instance, if you collect a large clinical data set, they'll have many empty spots if you've ever had the chance to work with it. And most of the time you do something called imputation, which is essentially algorithms that are designed to fill in those spots in a way that doesn't impact the conclusion. So it's meant to be relatively inert in filling those spots. And usually that's based on models of statistic. And so when those models approximate what to fill in or what to impute, they can also lead to increased doubts about the conclusions of the data. The old model of data generation used to be few companies are generating data and everybody else is consuming that data. But the new model is everybody's kind of generating data and all of us are also consuming data. We're starting to become the product, and if anybody's looked at any documentary with regards to like the social dilemma and stuff like that, we are starting to generate so much data that's being used to essentially sell to us all the time. The generation of data in terms of science and medicine, we're mainly referring to a generation of static imaging data like CT, MR, or histology slides, dynamic video data, animal behavior, cell migration data comes to mind. We have generation of static text data as well, like for example in large omics databases like RNA sequencing that we talked about, or proteomics or epigenomics, or even electronic health records like patient demographic data. We also generate dynamic text data like sensor data from electrophysiology recordings, respiratory readouts, etc. You often have to associate many of these with one another to gain more insight. So for instance, if you have a patient and you want to look at the genetic predisposition of an Asian male to respiratory complication after COVID, you'll see that you have to know, you have to get their genomic data, you have to get their demographic data, you have to get their comorbidity data, and all of these have to be chained together in order for you to answer this question. So there's two main challenges. The first challenge in big data is storage. And storage has been something that's been tackled quite well in terms of cloud storage and large server farms. So you look to Amazon and IBM and Microsoft to be providing these services. But that's not going to be our focus today. Our focus today is going to be the challenges that arrive with sort of analysis. So connecting and integrating these multiple different types of data, processing them in real time, integration of these into the cloud storage framework, which we won't really talk about, but it's something that's necessary in order for the analysis system to really do its job very well. We're in need of what we want to call an intelligence system that can function in a similar manner to humans. So when we talk about human intellect, we want to think about what makes us distinct. Evolutionary speaking, we have billions of years of evolution. We're the dominant species right now. We often learn from our mistakes, not all the time, but we do, and we learn. And we adapt ourselves to the environment in order to perform better. So these are the characteristics of human intellect. The definition of intelligence is to acquire and apply knowledge and skills. And artificial intelligence is then defined as the capability of a machine to imitate this sort of behavior that we see in humans. So AIs would use computers to model intelligent behavior with minimal human intervention. So ultimately, AI should be able to store, compute, and then learn from the data. So the general test that people have used in the past to sort of test whether or not a machine is intelligent has been referred to the Turing Test, named after the computer scientist Alan Turing, which played a pretty big role in decrypting World War II messages. So if you've seen what's that movie called? I can't remember what that movie was called. Imitation Game. There you go, with Benedict Cumberbatch. Exactly. So it was named after him, essentially, not Benedict, but Alan Turing. So essentially, how it works is you have a human questionnaire, which questions a computer respondent versus a human respondent, and you look at the number of times the human questionnaire is unable to distinguish the answers between these two respondents. So I want to show you a video of one case where people have thought that the Turing test was passed. And this was back, I believe, in 2018, when Google was demonstrating the capability of its assistant, its virtual AI assistant, which is now on your phones if you download Google Home or Google Assistant app, to be able to basically call in and make a reservation with a hairdresser. So I thought it was quite cool. Let me just see if this will actually play. Will this go? Let's see. Okay, so it refuses. Let me just go on YouTube and pull it up then, real quick. So, and can we get volume?
Haircut for a client. I'm looking for something on May 3rd.
So I'll just show it to you.
Oh, how can I help you? Hi, I'm calling to book a woman's haircut for a client. I'm looking for something on May 3rd. Sure, give me 1 second. Sure, what time are you looking for around? At 12 p.m. We do not have a 12 p.m. available. The closest we have to that is a 1:15. Do you have anything between 10 a.m. and 12 p.m.? Depending on what service she would like. What service is she looking for? Just a woman's haircut for now? Okay, we have a 10 o'clock. 10 a.m. is fine. Okay, what's your first name? The first name is Lisa? Okay, perfect. So I will see Lisa at 10 o'clock on May 3rd. Okay, great. Thanks, great. Have a great day. Bye.
So yeah, so they did a live demonstration, and they showed that essentially the human questionnaire there, which was the hairdresser, could not tell the difference between the assistant and a real human, and it was able to also contextualize the nuances of human speech, like when it did like just to kind of show that it understands that it has to wait, for instance. So to model intelligent behavior, researchers went and started looking at the brain, the architecture of the brain. And the brain functions as a result of an immense number of neurons that fire action potentials or signals. And the neurons connect through synapses that propagate electrical impulses by releasing neurotransmitters. And synaptic plasticity is the activity-dependent change in the effectiveness of synapses, or this is how we essentially learn, right? The synapses can alter the strength of their connections, and one principle of that is through what's called potentiation. So when one neuron consistently stimulates the other neuron, or one synapse consistently stimulates the other synapse, there's long-term potentiation that happens, there's habituation, and the neurons' connections become stronger. It takes less to activate them, essentially. And so neuroscience has been critical for the development of AI. The artificial neural network, or ANN, or we'll refer to them as neural networks for simplicity, mimics this aspect of biology by acquiring knowledge through learning, or what we call training in neural networks. It stores this knowledge by adjusting the weights within the network in a similar fashion to the strength of the connection. And a neuroscientist in 1949, Hebb, describes that if one neuron repeatedly stimulates a second neuron, the connection would strengthen. So the nomenclature for artificial intelligence kind of goes like this. You have artificial intelligence, which encompasses machine learning, and deep learning, if you've heard of it, is one subcategory of machine learning. So today we're going to be talking a lot about neural networks because it is one very complicated way of being able to mimic what the structure of a human brain is like to answer very complex questions. And we'll also talk about the different types of machine learning that we know today. So I'm going to give a quick crash course on neural nets. So our brain's pretty smart in the sense that you see these three handwritten images of 3, and you'll be able to easily distinguish them. All of these would be read as 3 in your brain. But if you were to tell a machine to do this, it'd be very challenging to do so, because what you would go is you would probably program the machine to interpret the white pixels in the black framework. So if you imagine every single pixel in that particular image is a grayscale value that goes from 0 to 1, 1 being white and 0 being black, you could potentially program a machine to recognize a certain pattern of white on a black background. But no way is it ever going to be able to distinguish these 3 using that sort of training. So the concept of a neural net is essentially you would take, for instance, in this case, a 28 by 28 pixel writing over here, and you would encode each one of these individual pixels, so it's 784 pixels in total, with a number from 0 to 1 that symbolizes its grayscale value. And we'll just call this for now the activation number, okay? So the neuron is just a thing that holds a number at this point. So the higher this number, the brighter the white. So in a neural net, and this is a diagrammatic illustration of what a neural net would look like, you have an input layer which contains the 784 neurons or nodes that encodes the picture that you're giving it. And you ultimately have the output, which you want it to be able to distinguish that it's not the number 9 in this particular case. So you can imagine that these 2 layers in the middle, and we call these hidden layers. And deep learning refers to the fact that there's more than one hidden layer. Okay, so in this case, this would be considered deep learning and not just simple machine learning. But in these hidden layers, and generally hidden layers are a black box, you don't really know what they're doing, but I like to conceptualize what they're doing for you. So the first layer here could be the detection of small little edges. So for instance, these different colors symbolize little edges that it could detect. So if you split this little handwriting into little blocks, and you ask the machine in each one of these circles here to light up when there is a little edge in that block, then you could get this pattern of lighting up to symbolize these various edges on this 9. This layer here could go further and take these edges. If they're lighting up, then that means that there's a line there. So you could go ahead and take this and assimilate it so that, for instance, there's a circle here if these neurons light up, and then there's a line there if these neurons light up. All right? And the presence of the circle and this line would then allow the machine to interpret that this is a 9. So how do we encode, say, in the first one of the neurons within that first hidden layer? How do we encode edges? So each neuron within the second layer has a combination called the activation function. And an activation function is essentially a series of weights and activation numbers. The activation numbers are the numbers associated to these 784 neurons that we get at the beginning. So you have A1 with the first neuron, A2 for the second. And each one of those activation functions also has a weight, as in how important is this particular neuron. So for example, this node, if we were to program it to, say, light up when there's an existence of one line right here, we would say, well, we want these pixels, okay, these select pixels here, to have a very positive weight because we want a white line to be seen there, all right? And we want the pixels at the top and bottom to have very negative weights because we want those to be empty, okay? And if you do have a pattern like that where all of these neurons are lighting up because there is a line there and all of these are dark, then this particular neuron will fire. This particular node will be activated. All right? Now, activation functions, if you can see from here, this sum would become a really large number. In order to squish larger numbers into a scale of 0 to 1, we apply a sigmoid function. And a sigmoid curve looks like this. So even negative numbers will become very small, and very large numbers would go towards 1. So using this sigmoid function and putting that at the front, we're able to squish all of this into a number from 0 to 1. Another characteristic of neural nets is this constant called the bias. So the bias essentially is there so that if this neuron is not all that important, we want it to only fire when the sum of all of these numbers is equal or greater than this bias. And so you add that there to kind of single out how important it is in this diagram that there is a line here. And that's an additional parameter that you can configure. So now in the hidden layers, each node or neuron is not just something that holds a number like the input layer. They're actual individual functions, right? They're very complex individual functions. So how do you learn with a neural net? So generally what we do with neural networks is we train them on labeled data sets. And we'll learn later that this is what we call supervised learning. When you have a data set where you have the handwritten message here and what we call ground truth, or what it's really symbolizing. So in this case, these are all correct. And we're teaching the network to make correct decisions by feeding it correct information. And then after it trains on this data set, we'll give it a separate data set called the testing data set, the validation data set, to see how well it actually performs after training on this one. So the most common data set people use for this particular application is called the Minced Data Set. And it's a large collection of these handwritten messages with the correct number that corresponds to them. So how learning works is you have to also tell the machine when it's making a mistake so that it can learn from these mistakes. So to teach the machine that there is a cost associated with the error, they designed things called the cost functions. So a cost function, essentially if you look at this one here, the machine is trying to look at 3, but it's having no idea whether or not it's 3 at all. All of these outputs are what it thinks it's supposed to be. So this is what we anticipate, but this is what it's giving. So this result's pretty trashy, right? If you wanted to penalize the machine for this, you would use a function in order to basically tell it, well, how wrong are you? And this is what a cost function does. And I don't need you to know the specifics of what a cost function is, but we want to understand kind of how complex these cost functions really are. So if you look at, in this particular example, 780-foot input layers, and you look at the number of weights and biases that are associated with all of the neurons in the hidden layers, you have a lot of weights and biases. You have 13,000 weights and biases with just this amount of input. And the output for the cost function is only one number. And this number, for instance, here, the cost for this mistake was 5.4. And what it would do is it would average the total cost for each one of these training sessions. And if your model was performing really poorly, your cost number, average cost, would be extremely high. So the purpose of training is to reduce the cost. The better the reduction in cost, the better your model is. So the concept of cost reduction, or essentially, is essentially the same thing as doing differential calculus. So for example, if this was the curve for cost, very complex curve, the cost is minimum right here, okay? In order for you to find the cost, generally speaking, you would be able to do differential calculus and just find the tangent, right? But in this case, we have multiple minimas. So doing tangential equations for a very complex cost function is sometimes impossible. So what they developed was something called gradient descent. And gradient descent is an algorithm that helps you find the minimums. And to understand gradient descent, essentially, you, at any point inside the curve, you could be up here, you would draw a tangent to the curve. And if the tangent was positive, if the slope of the tangent was positive, you'd move to the left. And if the tangent was negative, you move a little to the right. Now, if you keep doing this, you will eventually arrive at a local minimum. All right, so that's how gradient descent works. It's not easy to arrive at the global minimum, though. And that's one of the issues with training, is that oftentimes you can only reduce it so far as to find a local minimum. And the concept of stochastic gradient descent is essentially the randomization of your starting point. And if you randomize it many times, you'll eventually be able to find its global minimum. So a lot of these things are taken care of with iteration of training. You can imagine when we deal with a simple cost function that's only one-dimensional, we get this nice 2D plot. But when you're dealing with a cost function that is multivariate, then you'll get a 3-dimensional plot that looks like this. And in order to find the gradient descent, it's similar to rolling a ball down a hill. So you slowly roll it down, and eventually you'll find the path to the very bottom of the slopes. So what happens when you finally get these so-called gradient vectors? So a cost function will generate gradient vectors for all of the nodes inside your neural net. And these gradient vectors, all they're trying to do is tell the model how to adjust itself. So let's say it runs a simulation, it runs a training, and you get a cost function that's very large, and it computes the gradient vectors that resulted in that cost function. What the gradient vectors will tell you is you need to adjust some of these weights of neurons by a lot, or by a little, or decrease it by somewhat. It tells you how much you have to adjust the weights. You can't change their activation, but you can change the weights and biases. And by doing this, the next training will be so much more successful because of these adjustments in weights and biases within your model. So, and this refers to the concept of backpropagation. And what backpropagation is after you do a round of training, you would use the gradient vectors that you've generated, and you would go back and you would modify the weights and biases of these nodes. And by doing this in every round, it improves your chances at getting to a neural network that has a lower average cost. So when we do training, usually we do like 300 to 500 iterations most of the time in deep learning. And eventually we'll be able to have an overall performance metric that's usually like accuracy, precision of our model. And we'll also be able to look at the cost of our model, as in the loss, the cost functions. So just to summarize, the neural networks are made of layers of nodes that simulate neurons. The layers in the middle that exclude the input and output layer all carry an activation function that's composed of the sum of the activation, weights, and the biases from the nodes that feed into it. And all of this collectively determines whether or not that node is activated. Learning in the neural network requires training, and training errors have costs that are computed with a cost function. The goal of the training algorithm is to minimize the cost of these models. So in each iteration of the training, we have backpropagation that goes back and adjusts the weights and the biases based on the gradient vectors that we've generated, such that it has a better chance of reducing the overall error the next time around. And really, this summary is pretty much all I want you to take away from all of that math. All right? I'm not going to expect you to know all the equations and stuff, but the general concept of what a neural net is. So we're going to talk about the different types of machine learning and also a little bit about computer vision and how to apply these things in translational research. When we look at machine learning, we look at 3 different types. We have unsupervised learning, which we do all the time in statistics, and supervised learning, which we do a lot in machine learning and deep learning as well, and a reinforcement learning, which is this new concept that I'll show you. So unsupervised learning is when you're trying to find underlying patterns in the data, but the data is not labeled. And if you remember, we were talking about single-cell RNA sequencing the last time, which is a great example of this. You sequence a bunch of cells. You have no idea the identity of these cells. So most of the time what we do is we do clustering analysis to cluster the cells based on their transcriptomic profile in the hopes of finding their identities. And I'll show an example of an algorithm that's recently published that does that. So in this particular case, you could do it for patients in terms of precision medicine. So you could uncover a subset of patients who share similar clinical or molecular characteristics. So it doesn't always have to be transcriptomics. It could be multimodal, like you could look at grouping them by their certain parameters from their CT scan, et cetera. And you'll be able to say, well, there's 3 populations of these patients that respond differently to a drug, for instance. And that's what unsupervised learning is. And supervised learning is used to uncover the relationship between variables of interest or in one or more target outcomes. In supervised learning problems, the outcomes have to be known. So we're dealing with labeled data sets. So that example that I gave in the neural nets, that would be considered supervised learning because you are feeding it data when you already know what the ground truth is, as in you know what the right outcome is. And by doing that, the machine can then learn, well, what is the goal of its prediction? So if, for example, researchers want to know whether a set of clinical features, so vital signs laboratory tests, can predict ICU mortality, they can use a supervised learning algorithm where each patient record already has the outcome. So in the case of ICU mortality, you would already know if the patient survived or didn't survive. Then you would use that to train the algorithm to predict if you have a new patient with those set of features, well, what would the outcome be for that patient? Reinforcement learning, it's a different type of learning. It's kind of trying to put the machine inside an environment and giving it a set of rules that either rewards or punishes the machine for making errors or rewarding it for making the correct decision and letting it find out what's the best way to arrive. What's the best way for it to model the situation. So you would set certain parameters for reward and punishment, and the machine's goal in this particular style of learning is to maximize the cumulative reward. And one kind of example that it was applied to was gaming, because in gaming we have very defined punishments and rewards usually. So this is one such example. So these are Atari games. They're very simple games, and in this particular example, when you first exposed the machine to this, it was losing. And you could see that the points that it was gaining and the lives that it was losing, it could calculate the reward and the punishment. After about 30 minutes, it was able to perform better than a human that's been playing it for a very long time. And similar to this, same thing. Initial performance, and 30 minutes later, it was able to crush the game. So in environments where where reward and punishment is very easy to program, reinforcement learning does very well at solving these problems. Computer vision refers to the processing of an image in order to enable identification of an image input and provide an appropriate output. Most of the time, this has been applied to classification of images, and I just wanted to show you one sort of example of this. Let me just see if I can swap displays. Okay, So this is an online neural network, and I guess I have to switch it like this because I can't use it otherwise. Essentially, this is similar to the neural net that we were talking about, and it's an online simulation where this neural net's been programmed into a web interface. So what the goal is if I write a handwritten number, it should be able to tell me what that handwritten number is. So I'm going to give it a go. I'm just going to write a 9 and see if it does well enough to tell me what this is. So let me just zoom in. So I'm not sure if you could see it up there. Right up there at the thing. So it predicted on the last output layer that this was indeed a 9. Usually there is a little bit more. Let me see if I can get the values here from the source directly. So if you look at this here, it was able to take this 9, and it was able to get the first guess at 9 and a second guess at 1. This is one way to show you how this neural network breaks down that image. The reason why the second guess is 1 is probably because of the long stem that I drew. So if you look at the way we broke that down into edges and assimilated abstractions of the edges into lines, that's why we have these different guesses. But this is one example of what we call computer vision. And let me just switch this back. Swap displays. Okay. Another example of computer vision is Tesla's Autopilot program. Now, I'm not sure if this is going to play because I've been obviously having video issues all day long, but I'm just going to pull it up as well just to show you for image classification. So
Tesla Autopilot.
Wrong video. This one. So if you look at what Tesla Autopilot sees, it's able to discriminate multiple things. So it's able to discriminate the vehicles. It's able to discriminate the edges of the road, traffic signs, and you'll see even parameters like wet road, as in the probability of this road being actually wet, or whether or not it's raining. So this is real-time processing, obviously. So as you're driving, it's able to use a neural network to classify the objects that it's seeing. So that's kind of the ultimate application of a very complex neural network that's able to discriminate. And that was trained on millions of miles of driving from users. So there are other applications of AI that's relevant to translational research, and I'll just give a few examples of that. So cancer classification is one way we use AI these days. So classifications of tumors based on pathology or histology samples. We are recently publishing a paper using transcriptomic data to classify tumors. And we talked about using AI to classify cell clusters and labeling them in single-cell RNA sequencing, and using it to predict protein structures from polypeptide sequences. So in this first example, AI is capable of predicting these things called CUPs, and essentially CUPs are cancers with unknown primary. So cancers with unknown primary means that you don't know where this cancer came from or metastasized from potentially. So in this particular data set, the authors had a bunch of histology slides of different cancers that were stained using an HE stain, which is a basic histology stain to tell you whether a tissue is healthy or lesioned. So they fed this into a complex neural net, a convolutional neural net. We won't talk about what that is, but a neural network, and asked it to do two tasks. The first task was to predict whether or not this was a primary tumor or it was a tumor that metastasized based on its features. And the second task was telling it what kind or what origin was this tumor? Where is it from? And it was able to do it with very good accuracy. If you look at most neural network papers, the one graph that they'll really always show is performance, and this is called a confusion matrix. And what a confusion matrix essentially is, for example, if it's making a prediction that it is this organ, is that really the ground truth? So if you see a very neat line going like this, that means it's doing a very good job at predicting what the true label is. And this is, again, a supervised learning approach. And if you, I know that the colors aren't great here, but there are times that it made mistakes. There's some shades of blue here, somewhere in that chart. And oftentimes you also measure the receiver operating curve, so the area under the curve that tells you the sensitivity, as well as the specificity of your particular model. And you'd want that to be close to 1. And in both cases here, they're very close to 1. That tells you the model is quite accurate and good at predicting. So very, very good. No false positives and also no false negatives, essentially, is what these two graphs are trying to say. All right, and another case study here is AI predicting 3D protein structure. Now, this may seem like a trivial thing, but it's honestly been something that's challenged the field for a very long time. So you get an RNA sequence. You know that the RNA sequence produces a certain amino acid sequence, but determining the tertiary and quaternary structures of proteins has been notoriously difficult. Most of the time, you see a lot of studies do crystallography, and each crystallography study is like a paper because it takes so long in order to actually arrive at these very nice diagrams of what protein structures look like. And so why do we care about these things? Well, a lot of drugs interact with proteins in order to elicit a certain effect, and it does so by modifying active sites. So sites where, if a protein is an enzyme, for instance, where it could interact with the substrate, sometimes a drug. So in this case, this software called AlphaFold, based on a complex neural net, is able to take a polypeptide sequence with an unknown 3D structure and is able to render a 3D structure. And if you look at the green, it's the experimental crystallography structure, whereas the blue is what the AlphaFold ended up doing. You can see it matches extremely well in these three examples. As we talked about, so AI is also capable of determining the cell identities of single-cell RNA sequencing. So for instance, generally when we do single-cell RNA sequencing, we end up with a plot that looks like this. And this is what we call a t-SNE plot. And a t-SNE plot is a plot of all of the individual cells. Each one of those tiny little dots is a cell. And it reduces the dimensionality of the 17,000 genes that you've sequenced, and it tries to plot that onto a 2D graph like this. So generally what we do from this, without this software, is we do clustering analysis, which is an unsupervised approach, where we basically give colors to each one of these clusters. So now you'd be like, well, that's cluster 1, that's cluster 2, that's cluster 3. And then you'll say, well, that cluster, the most expressed, highly expressed gene is, for example, AIF1, which is a marker for macrophages, for instance. And you'll say, well, that's a macrophage population then. And you'll be surprised, you look at all these elegant single-cell studies before a lot of these software like this came along. All those labels that were put on the single cell were manual. Researchers just went in, looked at the top transcriptomic markers that expressed in that cluster, and just made a call and said, well, that is a macrophage, we think. Along came the kind of AI revolution. So we've been kind of going through, this is probably the third or fourth paper that came out doing something similar like this. Each one has pros and cons to it. But this one's particularly good at doing immune cells. It has a pretty large repository of highly expressed markers that discriminates each immune cell subpopulation from one another. So using this, it's able to do supervised learning and help you label the clusters. And it does so in what they call a agnostic way, where each individual cluster you believe is one discrete cell population. But it goes to do it in an extended way where if it thinks these two clusters are actually the same cell, for example, if they're both T cells, it can help you associate them to one another and identify them as potentially two heterogeneous subtypes of the same cell. So it's quite a powerful way at discriminating cells in single cell sequencing. And then recently, we have a paper that's in review right now where we take the transcriptomic profile of a tumor and we want to take the transcriptome and ask whether or not an AI can say, well, that transcriptome is from a healthy cell and that transcriptome is from a tumor cell. And we also got it to identify whether or not it was a cell that was just adjacent to a tumor, because it's been very difficult for clinicians to often identify what the edge of a tumor is. So when they're going in and they're resecting a tumor, it's often difficult to find where the boundary area is for where the tumor is, especially in the central nervous system. So we used a large data set of RNA sequencing data that's publicly available. They're all labeled, as in we knew which transcriptomes were from healthy tissue and which transcriptomes were from tumor tissue and which ones were from tissue adjacent to the tumor. And we designed a deep learning model where we have the input layer, 5 hidden layers, so it's a pretty complex model, and then 2 output classifiers. So it would tell us whether or not it was healthy, tumor, or tumor adjacent, and it would also tell us where the actual tumor was from. And then we designed a subtype classifier, which allowed us to identify the specific kind of tumor that it was. For example, a glioblastoma or a meningioma, for example, 2 different types of brain tumors. And I wanted to show you this particular outline of the architecture because I wanted you to appreciate all of the other parameters that goes into what a neural network is. So in each of these layers, we have different sets of neurons. And you'll be like, well, how do you know how many neurons to put into each layers? And the answer to that is we don't. And what you do is you have to actually do what we call hyperparameter tuning. And what hyperparameter tuning is you take this model, this architecture that you've designed here, and you literally ask it to change the number of neurons between 2 integer ranges. So in this case, the maximum number of input is the transcriptomic profile, which had 17,000 genes. So we had 17,000 neurons in this input file, in this input layer. But this first layer here, we tested it from 1 neuron all the way to 17,000 neurons. And we did the same thing for every single layer until it arrived at a structure or a structure of neuron layers that gave us the best performance. So everything is done by trial and error, essentially. And the way you do trial and error is you do something called hyperparameter tuning. So we did this is a confusion matrix that we plotted. So we were able to see, it was able to discriminate between tumor versus non-tumor cells with pretty good accuracy. This is our area under the curve. It almost basically reached 1 in most cases. And it was able to also discriminate most of the time which organ it was actually from. So this is our confusion matrix. Again, you're trying to look for this nice horizontal line. But it did make errors in several of them. These are misclassified errors, so you want to keep that to a minimum. And then you also plot the area under the curve. And at the end of these papers, you generally also have to compare it to state-of-the-art classifiers. So most of the classifiers, when we published this work, was done using traditional machine learning approaches, not neural nets. So things like vector machines, KNNs, k-nearest neighbor clustering. So we had to compare the performance of our model to those, and we were able to see that the deep learning model was superior to those.
And with that, I'd like to thank everybody for listening to these 2 lectures on the application of quantum computing and AI in medicine. I hope that I was able to give you a small primer on what quantum computing is and the application of AI outside of biomedical imaging, and also some of the healthcare implications that quantum machine learning can have. And I'd like to thank Dr. Pascal Tyrell for giving me the opportunity to prepare these 2 lectures for you. Your feedback on these 2 lectures is greatly appreciated. Thank you.